Individual fish within a population are typically not located randomly across the sea. They are spatially distributed in a dynamic way that changes over time and is influenced by many factors such as food availability, the presence of predators, and various environmental conditions. Changes in fish abundance is also associated with changes in spatial distribution. As fish become less abundant, their spatial distribution decreases. Understanding the distribution of a fish stock is important for ensuring that fishing is sustainable. If a population occupies a small area, they are more at risk of extinction, whereas maintaining healthy distribution helps to reduce the chances of overfishing. It's therefore important to understand and consider the spatial distribution of fish when giving advice on how much can be caught. However, the spatial dynamics of a fish stock are often ignored due to the complexity, assumptions, and data requirements associated with spatially explicit models. Instead of fitting a model, we can calculate indicators of a species distribution, such as their range, which measures how dispersed a population is in space, occupancy, which measures how many regions the population can be found in, and aggregation, which measures how clustered the fish are. To be able to use these indicators to inform how much fish should be caught, we first need to make sure that they can in fact tell us whether a fish stock is healthy or not. For fish stocks that have lots of good quality data available, we have a good understanding of their health status. We can then calculate our spatial indicators for these stocks and then compare the indicator predictions against the true known values of stock status. We can do this for many years' worth of data, comparing the predictions to the observations for each year. The number of years incorrectly and correctly classified can be used to plot a ROC curve. The area under this curve, the AUC, tells us how well the indicator performed. We use this method to test 10 spatial indicators using 52 time series of data. The AUCs varied, but they appeared to be quite high in many cases as indicated by the green sections in the heat map. To understand what influenced this variation, a regression tree was fitted. There was one group where all spatial indicators performed well, and another larger group where only the aggregation and occupancy indicators performed well. Overall, our findings show that the spatial indicators can indeed tell us about the health status of a fish stock, and this work identifies aggregation and occupancy indicators as useful candidates to be incorporated within fisheries management.